Hi everyone. Okay, this is bacterial reproduction and growth for this week's topic. Now, what I'm going to be talking about is not only the growth of bacteria uh, in a population, but also bacterial reproduction. So we do need carbon and energy for this processes. So there are general categories of growth based on their nutritional patterns. So we're worried about, or what I'm thinking about, is where's the ATP coming from? Where are the carbons coming from for reproduction? Okay, and we have two general categories: those that produce their own food, their own biomolecules. These are autotrophs, and those that need to consume already existing molecules. They need food from the environment, and those are heterotrophs. So anyhow, in the autotroph category, we have two subgroups. We've got your photoautotrophs and your chemoautotrophs. So I'm going to try the best I can to explain where the ATP is or comes from, and where the carbons come from with this group that makes their own food for reproduction. Anyhow, photoautotrophs capture the energy from the sun, okay? And then it is converted to that ATP. So that ATP then is used to pluck carbons from carbon dioxide and incorporate them into simple sugars. Okay, that's their food, the sugars. Okay, this process done by photoautotrophs is photosynthesis. Okay, in the chemoautotrophs, and bacteria do this, they don't get their energy or the ATP from the sun, but they get it from inorganic compounds. Okay, inorganic compounds. If you look at chemicals, chemistry, inorganic compounds, then they will take that inorganic compound and then convert that into ATP. And then the ATP from there is going to be used to pluck that carbon from carbon dioxide and make simple sugars. Okay? This process done by chemoautotrophs is known as chemosynthesis. Now, another category are those that have to eat food or consume already existing sugars, etc., and those are your heterotrophs. And we have two subgroups there. We've got the chemolithotrophs and the chemoorganotrophs. So the chemolithotrophs, another name for them is rock eaters, and only bacteria, note, only bacteria fall in this group. So what they end up doing is taking electrons, or the electron donors come from inorganic molecules such as hydrogen sulfide, ammonia, nitrate, and they'll take those and convert it into your ATP. But unlike the chemoautotrophs that can use that AT to make food, these guys don't. They basically will still need an organic molecule, organic stuff from the environment for their source of carbon. All right, finally, the chemoorganotrophs, which many bacteria fall in this group, including you and I fall in this group, they use molecules such as the sugars and fatty acids, amino acids, etc., as a source for both the carbon and the electrons for ATP. Okay, so this is kind of an overview of autotrophs and heterotrophs. Of course, there are some bacteria that can do all four of these. They're known as mixotrophs. And again, with them, they're very adaptable. It just depends on what's in the environment for them, where they are living, what is there for them to eat if they are heterotrophs. So anyhow, I just wanted you to appreciate that fact. Bacteria can do it all. All right, there's so many of them. They can do almost anything possible is possible for them. Okay. So what do cells do with the energy and biomolecules? Well, again, one of the things they can do is synthesize. Synthesize. And one of the processes of synthesis is reproduction, okay? Making more of themselves. So anyhow, usually bacteria will reproduce asexually by binary fission. And binary fission is actually a symmetric form of cell division. Asymmetric. 2 words I'm saying there. Okay, a symmetric form of cell division. And what that word means, symmetric, is basically you end up with 2 identical cells, same size, same shape, same genome. They're identical, okay? And how that happens is, the steps are right up here, you know, in the PowerPoint, is you've got your DNA replicating first. The DNA then will replicated DNA will start to separate into two sides of the bacterial cell. Now, bacterial cell is getting larger and longer in this process. And then your third step, you will have the cytoplasm starting to pinch off between the 2 potential cells. What is causing that is a special protein called FTSZ. Okay? So they kind of assemble to form a ring. In the center, usually in the center, especially for symmetric cell division, in the center of that bacterial cell. And they will start pinching in and separating the cytoplasm between the 2 ends of the bacteria. And at the end then you have complete division. A cell wall is laid down and you will end up again with 2 identical cells. Asymmetric cell division is just what it seems. You basically end up with 2 cells or more that are different sizes from the parent cell. And one way of asymmetric cell division is budding, where you have a parent cell that will develop the new cell. Usually it's extending from the original parent cell, and it's usually a smaller size, and it will break off at some point. Fragmentation is a cell, a bacterial cell, that basically is broken up, just as the word says, into different pieces after the replication of DNA, and each of those pieces then will go on to become a mature clone of each other. Bayocyte is just a type of cell that will produce many new smaller cells within it, and at some point they break out of the parent cell. Now, a sample of an asymmetric means of cell division is also endospore formation, and I will show that in the next slide. So I hope you know by now that endospore formation can happen in some gram-positive bacilli. Usually they're species of Bacillus or Clostridium. And what this is showing here is endospore formation. It shows binary fission, which is symmetric, but asymmetric sporulation is in diagram B here. So you basically start out with a parent cell, the mother cell, and the DNA replicates. That DNA then will either travel to one end or the other end of the mother cell. It will then form additional layers around that genome, a thick type of coating or coat, that will be a spore. And then the mother cell will die. That spore, as you know, can last many years, many years. And depending upon the environment, if it's good for that type of bacterium, it will then become vegetative. Okay, it will rise out of its sleep like Sleeping Beauty and become a living, breathing bacterial cell. Okay, germination occurs. Well, we do have another type of bacteria that reproduces in an endospore-like fashion. Okay, and that is in the bottom right-hand corner there. Epilopspiscium. Don't worry, you don't have to worry about it knowing the name of it or how to say it. But it's a giant gram-positive bacteria. It lives in a type of fish, a sturgeon. It is also a gram-positive bacilla-like looking bacterium, and it reproduces asymmetrically in a type of, like I said, endospore fashion. So what happens is a mother cell, once the DNA is replicated, will travel either to one or the other end or both ends of that mother cell. Again, what happens here is the offspring then will start to grow within the mother, okay? And at some point the mother dies, and then the new bacterial cells will leave the mother cell. But anyhow, it is kind of like an endospore-like fashion in that it usually, the new offspring are either at one pole or the other pole of the bacterial cell, which is similar to endospore formation in your Bacillus and Clostridium. Aren't bacteria great? They can do so many things. Okay, so we know bacterial cells reproduce by binary fission. And if the environment has unlimited resources, by that I mean it has a lot of nutrients for that bacteria, the pH is right, you're never going to run out of food, okay? Never have a buildup of waste products. It's like heaven, okay, for the bacterial cells. And what happens there is, if you have these unlimited, wonderful resources, the number of cells will double every generation. So within an amount of time, known as doubling time, they will double. So this is also known as generation time. So if you look at this, you've got one bacterial cell. Within a certain time, it's going to divide and produce two cells. Those two cells, within a certain amount of time, will each produce two more cells, and so on, and so on, and so on. But again, that time it takes for a bacteria to reproduce when you have wonderfully beautiful conditions can vary. And it can vary upon the bacterial species and also those growth conditions, okay? So the bacterial species, if you see here, the first one is Escherichia coli, and it has wonderful medium that it just loves with glucose and salts. And if you've got a good temperature, you've got that food there, it will double in 17 minutes. Or the generation time is 17 minutes to produce a new generation. And if you go down there, you'll see it really depends upon the type of bacteria and their nutrients, if they've been met, as to how long it takes for them to double or create a new generation. If you go down there at the bottom, second from the bottom, Mycobacterium tuberculosis, it's got a very special medium that they throw in some malachite green and special amino acids, glycerol, potato starch, even coagulated eggs in order to grow this guy. He's a slow grower. If you look at that, it takes between almost 800 to 900 minutes for that bacterium, one bacterium, to reproduce. And that's because, first of all, he's got a slow metabolism. You know how some of us seem to say we've got a slower metabolism than someone else. Well, bacteria too. He's kind of slow on the go here, his metabolism. And he also has a capsule, which takes a little extra time to make. He also has that special cell wall that has that mycolic acid in it. Okay, this is your acid fast bacterium. So he's got some extra stuff there he's got to make. It does take longer because of that extra stuff he has. So just appreciate the fact that one bacterium is not the same as others, and one will not replicate the same amount of time another one may. Okay? Okay. Now let's say we've got bacteria that are multiplying at a regular time. Okay? You're going to be increasing that number exponentially. And during this time, when they're multiplying at their best, that is known as the log phase growth. Okay? Now, there are two graphs here. And they're both kind of saying the same thing. Except the graph on the right is done in the log of 10. So what that means is, basically, you've taken everything on the left, the number of bacteria, you've graphed them, but it's been converted into numbers that kind of will condense the data, okay? So that we can see it in graph form, in an easy graph form. So for example, once we put all this data together, and it's in this log 10 log phase growth scale, we are able to determine that if a bacterial population is under a controlled environment, in other words, they only have so much nutrients. They only have, it's at a good pH, but they're only given so much food. We are able to see over time, using this type of graph on the right, how a population of bacteria grow and die. So this is known here as a bacterial growth curve. So again, it's got limited resources. It kind of will take a population of bacteria, condense it into a form in which we can see where it's present but not quite reproducing yet, where it's reproducing exponentially. Those generations are being produced in a regular, direct manner. Then we get a stage where maybe, they aren't dividing as much. They're starting to die because they're running out of nutrients. They're running out of food. And toxins are building up. And then finally we can see the death or decline phase of a population of bacteria. So if you think way back when you first started your skin project, and if you saw that plate now, if you still have them in the lab, you will notice that original plate, the agar is dried out. It'll probably look like cellophane. And you've got bacterial colonies that, they don't look the same. They're kind of crusty. They aren't vibrant. They're dying. They're dead because they've run out of the food in the media has dried up and they've used all that food up. So this is kind of putting that idea to a graph. So anyhow, we've got 4 stages. The first one is your lag phase. So you've introduced a bacterium into a medium, a liquid medium maybe. And what we see is basically there's not a lot of replication going on. These bacterium are getting used to the new environment. So they're maybe growing, they're metabolizing, but they aren't reproducing yet. So that lag phase looks kind of like that first that first line there, kind of a flat line, okay? Then, once they get acclimated to their surroundings, the population will go into a log phase. Okay, again, exponentially increase in number of living cells. They are reproducing at their best here. If you have an E. coli, every 17 minutes, that population is growing beautifully. And this is where usually when they're testing antibiotics, they will pluck bacteria in this stage of growth in a population. Next, we go to the stationary phase, and this kind of plateaus out. That's number 3. You've got bacterial cells, but again, cell division and those cells dying are roughly equal. And why is that? It's because they are running out of food. Also, because of reproduction, because of the metabolites, the end products of metabolism may be toxic. There's more toxins also building up in the media. Finally, we have stage or phase 4, the death or decline phase. This is an exponential decrease. So you've got basically more cells dying than reproducing. Okay? You should know these 4 stages and what happens in each for an exam and be able to identify each stage. Okay? So finally, we can figure out the number of bacteria that are in a population if we know the number of generations. So for example, let's say we have one cell. I'll read this out. Escherichia coli has been shown in experiments to have a doubling time of 20 minutes. So every 20 minutes, Escherichia coli is dividing. For a culture of E. coli that you started with only one cell, how many cells will there be if you let the culture incubate for 6 hours? So if we look at that problem, we're going to look, first of all, we need to know how many generations have been made within that 6 hours, okay? Before we can find out the number of cells that will be at the end of that. So anyhow, we know 6 hours, we've got 6 hours. Each the bacteria reproduces within 20 minutes. So you're going to have 3 generations per hour, okay? They're replicating every 20 minutes, and they're 20, 40, 60 minutes. So 3 generations within an hour. And we have 6 hours, so you'll be multiplying 3 by 6, okay? There we go. 3 generations per hour. Multiply that by 6. We get 18 generations of E. coli within 6 hours. We're not done yet because we want to know the number of cells. So what we do is we plug that 18 as an exponent to 2. N, the capital letter N, means the number of cells. So we plug 18 in there, as you see, and N will be 262,144 cells of E. coli in a population after 6 hours. So after 6 hours, starting with 1 cell, we will have over almost close to 300,000 bacterial cells of E. coli. So what does this mean? How does this affect us? Well, if you want to look at it in a practical way, at lunchtime you had 1 E. coli in your undercooked burger. If you left it out on the shelf or on the counter, at dinner time you'll have 250,000 of E. coli in your GI tract. By breakfast the next day there will be billions if you still decide to eat that hamburger, and you will be pretty sick, most likely. Okay? Okay, now in the real world, bacteria don't usually grow in and of themselves by themselves. What they end up doing is growing in populations or communities which are known as biofilms. So, how that works is basically you've got one or several cells of free bacteria. They're planktonic, another name for free floating or free swimming bacteria. These bacteria will be motile, is known as planktonic bacteria. And then they will adhere, stick to an inanimate surface usually. Now you can find these in pipelines, you can find them in nature, you can find them in us. Especially they're finding out now these are related to disease. So you end up having a biofilm. It begins with some free cells attaching to an inanimate surface. You also must have water available for these bacteria. And once they attach, they usually take seconds, okay, for them to attach. And it's kind of a weak attachment. It can leave at any time. At this point, they may, they may stick, they may not, depending on whether or not they find things favorable. Then you will have, at some point, maybe seconds or minutes after that, they will become irreversibly attached. They are stuck there permanently. The cells then will begin to grow and divide, adding to that biofilm. Now, it may be one type of bacteria, it may even be several types of bacteria that will adhere, grow, and divide. So, as you're going along, you're getting more and more added to this community. And at some point, step 4, you're going to have almost like a tower form. It will have water channels because, again, you need water in order for the nutrients to come and the waste to leave that little community. And at some point, number 5, they don't quite know why, but the biofilm, maybe it runs out of nutrients or whatever, you will have free bacterial cells, or those planktonic cells, break away from that biofilm in order to establish a new biofilm. That's it in a nutshell. I'm going to end with this quorum sensing. What they have found out with biofilms, bacteria within biofilms, is that individually bacteria may give off these chemical signals. Okay, little chemicals or molecules. If there aren't other bacteria around, those signals will just go off. They pretty much disappear into the environment. But once you start getting an establishment of a community, of a biofilm, you get more and more bacteria being produced, more and more bacteria coming into the picture. You'll have more and more of these chemicals being produced. At some point, that biofilm will reach a level where they have produced enough cells, or enough cells are present, enough chemical signals are being released that will affect the behavior of that bacteria, or that bacterial group, or that biofilm. And a case for this would be, certain genes will be switched on, such as toxins, okay? The gene to make a toxin by a bacterium may not be switched on if you have 1 or 2 bacterial cells, but you get a bunch of them in a biofilm. Those signals, that's called quorum sensing, those signals will finally reach get to the bacterial cells and will switch on that gene to make toxins, which then will be released and affect us if they are within us, within a, so do you see that by and large the bacterial films are pretty much antibiotic resistant because of their makeup, because of the bacterial cells they have, because of the quorum sensing. So anyhow, I do want to end with this. There are 2 videos here. Definitely look at the Bonnie Brazier one. You can directly link into this from the slide. I'm also going to put the one up here with Dr. Garth Ehrlich. This is a 6-minute montage. It's interviewing doctors and the effects of biofilms and disease, okay? And then also at the end of that one video with Dr. Garth Ehrlich, it gives you a nice little summary, a nice little animation of biofilm formation. So anyhow, I'm going to end here for today, and I hope you all have a good day, and I will have part 2 soon for this week. Okay? Thank you.